This presentation is titled From Field to Table, Chronic Wasting Disease and You. So what I'm going to talk to you about today is a bit of information around what it would mean for if chronic wasting disease did jump that species barrier, but I'm going to give you some background and some information to help you make your own decisions regarding chronic wasting disease and any potential risk to humans. So firstly, I would also just like to acknowledge that I am calling in from the unceded territories of the Coast Salish peoples, so namely the Musqueam, Squamish and Tsleil-Waututh First Nations. And I would also like to acknowledge the Tanaha First Nation on whose lands chronic wasting disease is unfortunately present at the moment. And big shout out to them and their strength and cooperation during this time. So awesome. So what we're going to talk about today, I'll give you a bit of an introduction. So what is a prion? What is the thing that causes chronic wasting disease? I'm going to talk to you in general about prion diseases, symptoms, and how they are transmitted, both in other animals and in humans. The species barrier, what is it and why is it important? And then chronic wasting disease and the risk for humans. Is it a real risk? Is it a perceived risk? But is it worth taking the risk? That's the question we're going to ask. And then some information on how you can protect yourself as a hunter. Just so we're all aware, I will be going through this presentation and then afterwards there will be a question and answer whole session and a discussion. So if you've got any questions, feel free to just note them down and we can cover all of them, hopefully after the presentation. Awesome. So let's start from the beginning. So what is a prion? So a prion is a protein. And on the top right here, we have a normal prion protein, and it looks like a protein. Normally, a prion protein is described or written down as PRPC. So this little tiny C denotes common, which implies that it's in its normal form. And prion proteins are very well conserved in mammals, which means that they are present in lots of different mammal species, from rodents to primates, which tends to imply that they have a lot of important features that are conserved during evolution. So what do? They aid in sleep regulation. They aid in the development of neurons, cell communications, stem cell renewal, cell signaling, cell adhesion, a whole lot of different functions within the cell. And they are highly concentrated on the end of nerves. These proteins are encoded by a specific gene, and I've just noted here the types of variations that this gene can have. You don't need to remember met or val as a variation, but it's just important to note that there are variations in the gene. An important thing about a prion is that it can misfold, and it does this spontaneously. And we don't necessarily know within the scientific community why it happens spontaneously. There may be some genetic predispositions or vulnerabilities caused by some of those met or val variations. And one of the issues with this misfolding is that misfolded prions, which are denoted as PRPSC, cause normal prion proteins to misfold as well. So what we get is this protein on the top right on the left here, the normal one, looks normal. It will randomly, out of nowhere, sometimes create this big spirally red thing. That is the misfolded or diseased prion. And what you can do is just kind of envision that a normal prion is a slinky. It works like a normal slinky. You can pull it apart, you can roll it down the stairs, whatever you want to do. But when it's misfolded, it creates this big, awful tangle. And that big, awful tangle means it cannot operate in the way that it normally does to help all of those key functions and sleep and development of neurons, etc. So what does that mean? Well, we end up with, up the top here, a normal structure of prion proteins, a lot of normal slinkies. They're just doing their functions. And what happens is one prion protein will misfold into that janky looking kind of slinky. And when it touches or interacts with another prion protein, it causes that one to then misfold. And then it touches another and another and another. And in a chain reaction, it causes a number of these prion proteins to misfold. So why is that important? Well, they do exist along nerves. So where are there a lot of nerves? In the nervous system, of which your brain is a very important component. And what happens is we end up with a big clump of these misfolded prion proteins that cannot operate like a normal prion protein. So we end up with gaps or little spongy looking areas in brain or nervous tissue, and that causes issues. So we've got a healthy sort of section of a healthy brain tissue on the left, and on here on the right we have what is a diseased prion, diseased brain tissue slide. So as you can see, there are a number of little holes in here. So that's not great, obviously, and this causes a number of prion diseases. So the types of diseases that these are called are transmissible spongiform encephalopathies. So that means transmissible, it can be passed from one individual to another. Spongiform, it makes that sponge shape in the brain or nervous tissue. And encephalopathy implies issues with the brain in terms of behavior or activity. So what you might see in certain Alzheimer's or dementia is also a type of encephalopathy. So this is not great, obviously. And what you find is that prion diseases can either be spontaneous, as we've talked about, genetic, so that is you have a genetic predisposition to have misfolding, or it's infectious. So one of the issues with prions, because they are so well conserved, as mentioned, is that we have a number of different prion diseases that affect a number of different animals. And this is not just limited to deer. There are a number across a number of different mammals. So for instance, we've got scrapie in sheep and goats, and that is where that original PRPSC comes from, because it stands for scrapie. We've also, it affects minks, it affects cervids and chronic wasting disease. One of the most important is bovine or mad cow. Oh, I think someone's got their sound on. I'm not sure if everyone can hear me. Okay, thank you very much. So bovine spongiform encephalopathy affects cattle. No, this is what was called mad cow disease in the UK outbreak in the 90s, what people have probably heard of the most. It can also affect domestic cats. There is an exotic ungulate encephalopathy, which affects nyala and kudu, and also in camels. So there are a number of different species that chronic wasting, prion diseases affect, and of concern to us is chronic wasting disease at the moment. So for those that don't know, and you probably all do, but I will just give everyone a brief overview of chronic wasting disease just in case. So it is a prion disease of cervids. So it affects white and black-tailed deer, mule deer, elk, moose, caribou. There are some cervids where it does not impact, but for BC and most of North America, these are the most concerning animals or species that are impacted. When a animal is infected with chronic wasting disease, we do get those holes in the brain that you can see under special lab equipment, but what we also see is other impacts. So symptoms that are clinically available that you can see with your own eyes. That includes wasting or weight loss, hence chronic wasting disease, confusion and unsteady gait. Chronic wasting disease infected animals are more likely to go into cars or roads and be hit by cars, etc. They also have an unsteady gait, extreme hunger and thirst, so they eat a lot, they drink a lot, and they produce an excessive amount of saliva when they're in a late stage of disease. Now, one of the things that Amber Lee mentioned is that the incubation period for chronic wasting disease in a deer is really long. So it is between 16 and 36 months. So that means between 16 and 36 months, the animal looks completely fine. There are no obvious symptoms, and they are actually getting sicker and sicker and sicker. And during this time, while it's a silent carrier, we call them, they can shed prions into the environment. So prions, those proteins that if they touch a healthy protein, they can cause more infection. So that's not good. What we find with chronic wasting disease is animals have a reduced lifespan, both indirectly and indirectly. So the time from oral infection to death in mule deer is about 13 months, so about a year. And what that means is it can take 36 months, so 3 years, for an animal to show infection, and it can die within a year. So lifespans are directly infected by the disease, and 3 to 4 months after symptoms appear, 34 months after symptoms appear, the likelihood of death is increased significantly. So infected deer in general are 4.5 times more likely to die annually than uninfected animals when picked at random. They are more vulnerable to predation, they're more vulnerable to car strike. And one of the issues with that is you end up with overall a reduced lifespan of the herd. You end up with population declines in areas, reduced herd fertility and recruitment, so that is fewer babies are born, and fewer fawns grow up to be of reproductive age and reproduce successfully. Additionally, we also have population dynamic shifts because male deer are more likely to be infected than female deer. And as an example of this, just down the bottom here is some annual adult deer survival statistics from 2023 in Wisconsin. So when uninfected, on average, year to year, a female random deer picked at random that is uninfected has an 83% likelihood of surviving until the next year. In males, that's about 70%, 69%. However, when they are infected with chronic wasting disease, we find that that drops to 41% for females and 17% for males. So chronic wasting disease significantly impact how long and the likelihood of survival of these cervids. So the real question that we want to ask, though, is what is a transmissible spongiform encephalopathy in humans, now that we know what they are in deer and other cervids? So there are a number of TSEs in humans, and I'm going to say TSEs because it's quite a mouthful to say the entire phrase. So one of the ones that you may have heard of the most is Creutzfeldt-Jakob disease. So this is a disease that also causes that holy appearance in brains or spongy appearance. And as you can see along the top here, the A, B, and C, these are cranial images of a brain scan showing a decrease in brain mass over time. So in general, Creutzfeldt-Jakob disease, most of it is spontaneous. So 85% of all cases known have been caused by spontaneous, random prion proteins misfolding. 5 to 10% of that is a genetic mutation, where you are more likely to be predisposed if you have this mutation. And it affects about 1 in 1 million people annually. So it's pretty rare. It's often mistaken for Alzheimer's dementia. It has very similar clinically presenting symptoms, and generally the onset age is around a similar-ish age for Alzheimer's dementia of being over 60. So there could be more people that are positive for Creutzfeldt-Jakob disease without being identified or diagnosed with it specifically. What I will jump to is these ones on the right. These are more less common. So kudu, you may have heard of. So kudu is infectious chronic Creutzfeldt-Jakob disease, which occurred via cannibalistic activity in Papua New Guinea. And that it's estimated that Creutzfeldt-Jakob disease was spontaneously originated in somebody who then was cannibalized due to a funerary procession cultural activity, and it was spread through there. So that is infectious CJD. We have GSS syndrome, which I'm not even going to try and pronounce because I will butcher the German words, so I apologize for that. This one is genetic. It's extremely rare. There is fatal familial insomnia, which is also genetic. And when we say extremely rare, I mean there are 70 families worldwide. Very, very rare, also caused by those prion proteins misfolding. And then VPSPR, which is an even more rare protease-sensitive prionopathy, which means it's complicated, but it is sensitive to different types of proteases. So has a similar set of clinical symptoms. But the one that people are most concerned about when thinking about chronic wasting disease is variant Creutzfeldt-Jakob disease. And you may have heard of this because this is the technical name for mad cow disease. So it was linked with consumption of beef products with bovine spongiform encephalopathy prions. So that is cows that had mad cow disease, or the prion disease that affects cattle, being eaten by people. So it's estimated or assumed that cows would have got BSE originally from being fed meat and bone meal, which was to give them more protein, that was potentially contaminated from either a cow that spontaneously got BSE or perhaps a sheep that had scrapie, which is the TSE prion disease that affects sheep and goats. So what happened is that 178 cases were found in the UK of the CJD, or variant Creutzfeldt-Jakob disease, and then 50 cases were found internationally due to an outbreak of this in the 1990s. It is estimated that there is genetic susceptibility here, so that certain people with those different variations in that gene may have been more susceptible. The onset of this was around 30 years old of age, which in comparison to normal, random, or spontaneous Creutzfeldt-Jakob disease is significantly earlier. So the symptoms of TSEs in humans. So as we talked about earlier, holes in the central nervous system tissue cause neurodegenerative disease. I do have to note it is always fatal. There is no cure. Same with chronic wasting disease and cervids. And these all have a very long silent incubation period in humans. So CJD, the one that is more spontaneous, tends to be dependent on route and how it gets to the body. So that could be, for instance, either through being spontaneous, if you were to have a blood transfusion with prion positive blood, so medical intravenous contamination. It can change the speed at which that it impacts you to get symptoms. But in general, it's around about 10 years. It's very important to note with variant CJD, so this is via cattle consumption or beef consumption, it can take between 10 to 20 years to become symptomatic. So that's a long time. You eating some beef when you were 20, and at 40, you get positive symptoms for variant CJD. Now with CJD, variant CJD, medical transfusion, so that's, for instance, contaminated blood transfusions, for example, is a bit quicker, between 6 to 8 years. However, while it may take a long time to become symptomatic, death is quick once you become symptomatic. So within normal CJD, 70% of people die within a year of diagnosis. So it might take a long time for CJD to make enough damage within the brain to become symptomatic, but you'll often, 70% of people will die within a year. For variant CJD, so that's the mad cow one, it's around about 13 months, so around about a year. Again, can take 10 to 20 years to become symptomatic, but you're dead within the year. So not ideal. In general, TSEs across the range of different TSEs and species, there are species-specific symptoms. As you noticed with chronic wasting disease infected cervids tend to have hyperphagy, which means they get really, really, really hungry, really, really thirsty. For instance, that's not something that we know about in TSEs in humans. In general, what we do find, balance and coordination is impacted, mental incapacitation, convulsions, tremors, and shakings, and then also complications, including aspiration pneumonia, which is caused by inhaling food or liquid when trying to eat or swallow due to issues with the muscles working. So, as we've talked about briefly, transmission of proteins, so genetic, infectious, or spontaneous, talked about that a little bit earlier. And with CWD, it's the infectious route that we are worried about. So within deer or within other cervids, prions move from the digestive system. So for instance, a deer might lick the ground, or they might have a conspecific relationship with another individual of their herd, for instance. And that enters the digestive system. It goes then into the lymph nodes, and from the lymph nodes into the nervous system, where they start to accumulate. Over time, as deer get more and more infected and the prion load within their bodies increases, there is spillover to skeletal muscle tissue. This is often excreted as well in body fluids in the environment. So this diagram down here is a really good summary. So we end up with marking, grooming, and mating. So an infected animal will infect other animals within the herd or within other herds. They will then urine or feces will be contaminated with prions as well. That goes into the soil. Another animal touches the soil, gets infected again. Once that animal eventually dies of chronic with or of chronic wasting disease, those prions are then leached into the soil over time, which then increases that environmental load. One thing that's really important to note is that prions are not destroyed by cooking or processing of meat. So this is particularly important when related to skeletal muscle tissue that you might eat. So last year, or earlier this year, I should say, chronic wasting disease prions were found in raw, processed, and cooked elk meat from Texas, as well as chronic wasting disease in 2024 was detected in white-tailed deer as well. So what this means is that if you eat chronic wasting disease positive meat, you are likely eating chronic wasting disease prions, which you might say, oh my gosh, that's terrible. Am I going to get Creutzfeldt-Jakob disease, variant Creutzfeldt-Jakob? We don't know. So, and this is because of something called the species barrier. And in order to explain the species barrier, I will very quickly take you through some very basic sort of biological evolutionary thought process. So species and their pathogens, so we've got one on the left and one on the right, evolve adaptations and counter adaptations constantly over their evolution. So for instance, you might, if you envision a pathogen that tries to attack your respiratory system, you have evolved coughing. So then the pathogen will have to evolve a different way to enter the body, for example. Or if we think in a more general terms, I have evolved a sword and I'm going to try and stab you, you have then counter-evolved a shield. So now I have to find another evolutionary way to get around your shield. And evolution tends to just go back and forth like this, and it makes many pathogens evolve to be very specific to their hosts because that one specific pathogen has evolved a way to get around that shield. So that's great. Generally, it does mean that the jump from one species to another is difficult. However, there is something called an intermediary species. So passaging, so having the nasty pathogen be in another species can cause it to mutate more quickly because it's a different environment. That means that those pathogens can become more human ready. They've got different evolutions that can then cause that species barrier to be less impactful. And this diagram here sort of shows a really basic way of looking into this. So we've got shedding of prion proteins on the left, going through into the environment. The species barrier, it may be picked up by a number of species, and as it evolves in there, it might make its jump to humans, which is something we don't know yet. There are a number of species that can, via experimentation, and that is direct contamination or injection with chronic wasting disease prions, can carry it, such as mustelids can, some rodents can, squirrel monkeys can, some ruminants can, but there are also a number of species that cannot. And particularly important is macaques, so non-human primates have not been found to carry chronic wasting disease under experimentary environments within a laboratory setting. So the big question that we have to sort of ask is, can chronic wasting disease jump the species barrier? There's research on both sides of this that say yes and that say no. So for instance, you may have seen this article that came out last year where 2 hunters from the same lodge were afflicted with sporadic CWD, and the authors asked, is chronic wasting disease to blame? So there has been to date no causal link found between chronic wasting disease, infected meat, and human prion-related illness. But we don't know that. So the question then has to become, is it worth the risk? So variant Creutzfeldt-Jakob disease from eating contaminated beef can take up to 10 years to become symptomatic. Once it's symptomatic, patients can die within 13 months, about a year. And at this point, we cannot rule out that chronic wasting disease prions may cause disease in humans. We don't know. And I will be completely honest, science doesn't know this either way, yes or no. But my advice personally would be to not eat cervid meat hunted in the management zone until you've tested it. So Kate has been working very hard with the province to get a sample testing turnaround time within the main management area between 2 to 4 weeks. And that is a goal, and that's amazing. So the question really does have to become, is waiting 2 to 4 weeks worth it? In my opinion, yes, but it's up to you. You are individuals with your own decision-making capability. Additionally, sampling and testing of animals also aids in surveillance efforts. For instance, where is chronic wasting disease and how prevalent is it? So my advice in general would be to keep hunting, keep testing, and keep healthy. And one final slide that I will take you through in a second is how to protect yourself, some basic biosecurity. There is a lot of advice online around how to submit deer heads for testing or lymph nodes. And there are a number of freezer drop-off areas across BC, which is in that bottom right map. There are also some further up in the northern BC, which I have had to crop here. So there are some scattered across the province, and that is all available on the Hunters for BC website, but then also on the provincial Waterland and Resource Stewardship website as well. So general, how to protect yourself. So just some advice. While hunting, avoid harvesting sick-looking animals. If you see an animal that is skin and bone, it's wandering around, please don't hunt it and eat it. That's probably not the best idea. But call the local hotline. So get the wildlife biologist, the wildlife veterinarian, and alert them or let them know. Information on how to contact them is on the chronic wasting disease website, both the provincial and Hunters for BC. After your hunt, when you're field dressing, preferably, if you can, separate your tool sets. So remove high-risk tissues with one set and edible parts with another. Wear disposable PPE, so that is gloves, eye protection, and aprons during processing, because you don't want to get blood in your eye that could possibly have chronic wasting disease prions in it. If you can, again, if possible, avoid using the same equipment in the management zone and non-management zone areas. So in general, my advice would be to assume that meat harvested in the management zone is positive until a negative test result is received. That's the best way to play it safe. Leave high-risk tissues in the field. That includes spinal column and brain tissue. Do not transport brain tissue or spinal column into new areas. Please, 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 please don't do that. As mentioned, submit your cervid head or sample for testing. And then when you get home or you're able to do it in the field, please clean and disinfect your equipment. So that is scrub equipment with soap and water to remove all organic matter, and then sanitize with a strong bleach solution and let sit for 60 minutes. This is known to get rid of chronic wasting disease, and there's a number of online bleach concentration calculators that you can use in order to make sure that you've diluted your bleach properly. But yeah, keep hunting, keep testing, and keep healthy would be my advice. And that's it from me, and I will stop sharing my screen.
Well, that was awesome. Thank you. The bleach concentrator thing, I'm gonna have to look that up because I never actually knew of such a thing. I kind of dump it in and then I go. So we are going to open this up to the floor. And a reminder for my participants, we also have Kate Nelson that's with us tonight too, our biologist champion down in Nanaimo, our wildlife health biologist for CWD. So she is here to support and to answer questions and all of that sort of stuff. So if those of you are familiar, you have a react button either on your iPhone or your desktop where you can put your hand up. I think it's the react button. Yep, it's the react button. So raise your hand. You can type a question into the chat and I can call your name out. So I'll give you guys a few minutes to think through that. I have something that has occurred to me for a little while. I never really considered a protein as being dangerous. Like I learned in elementary school, complete proteins and you're supposed to eat, a variety and all of that kind of stuff. But I have always thought of, bacteria and viruses, but a protein, like it just seems like such a benign, weird thing to be contagious. So that was my shock for the night. In one of your slides, you showed males with CWD and or bucks and does, and the does were 41% in any given year likelihood to die with CWD, and the bucks were 17%. Any thoughts on why that difference between the bucks and the does?
Yeah, this is also something Kate might be able to comment on as well, as biologist, but uh, it's not known specifically why, so we can't tell you in a sentence. But it is suspected that males have different types of behavior. So we assume it's a behavioral difference between males and females. So males tend to interact with other males outside of their herd a lot more than females, for example. So they're more exposed. They're also doing different activities that make them more likely to, I guess, touch other males that have chronic wasting disease, and also a lot of their behavior around marking of territory and other sort of very male macho deer behaviors can just cause an increase in likelihood. But Kate, if I've left anything out there, please jump in.
No, that's great, Jessica. Can I just start off by just giving Jess a shout out because that was just such a great presentation. And, I think about this stuff all the time, but I know a very little, a bit about a bunch of different things. And to have an expert like Jess with her background and just depth of knowledge with this stuff has just been invaluable to the program. So thanks, Hunters for BC for pulling her in, and it's great to hear that. So yeah, in this context, the short answer why males are generally more likely to, infected males have that increased rate of mortality, we don't know. It's complicated. There's a lot of different factors, like Jess alluded to, behavioral things that would make them more vulnerable. It could also be something to do with, yeah, their exposure to prions and sort of that prion load within their body that might increase the rate of mortality in those animals. Yeah, any number of things. But yeah, it's likely very complicated. We can kind of, point to some likely variables, but we really don't know.
Awesome. David.
Hi, Jess. At the start of the presentation, you listed some other animal prion diseases that affected different species. Have any of those ones been shown to jump the species barrier to humans? Or is it just BSC?
Currently, no. There's been no other evidence. That being said, to be totally honest, no one's looking. So we know that, for instance, the domestic cat spongiform encephalopathy that was from BSC related, so cattle meat infected with bovine spongiform encephalopathy. So at the moment, no one has found any evidence. It's not known that scrapie in sheep or goats either jumps to humans. But I would have to look into, in a bit more detail, the likelihood with scrapie. I think, to be totally honest, it was only very recently, so in the 90s, that prions became an interesting thing to study. So mad cow disease put a lot of money into prion disease research, but before then, there was nothing. So Amberly, as you mentioned, people didn't really consider prions as a protein to be an infectious agent. It wasn't something that anyone had ever heard of. In fact, when that first hypothesis came out that it could be a protein, people laughed. People were like, it can't be a protein. That's impossible. So at this stage, we know that the bovine TSC is an issue for humans. Trying to find the causal link there is really complicated. So if you know any epidemiologists, you might know that trying to find the statistics that correlate that can be easy, but then finding the mechanism to prove that it was the meat that caused the symptom, especially when that incubation time is so long. And the way that they did that with bovine encephalopathy was looking at very specific, teeny tiny genetic coding regions of the prion coding gene in humans, and then samples of meat, as well as looking at different pieces of information within the appendix of a number of people. So it was really, really hard. And I think when they first came out with that, again, thought of, oh, could this be related to BSE meat? That was also considered very skeptically. So until there is more focus put on that, I don't know if we would find the answer, especially for the other animals that we don't eat and we don't farm. Yeah. So the answer, unfortunately, is we don't know.
That's great. Thank you.
So are we actually going to be looking now for like human related, prion disease from CWD if it's that hard to even trace in BSE?
So there is a lot more research coming into it, because humans are, just to put us all the way back, we're a little bit selfish. If it doesn't impact us, we don't really tend to worry about it all that much. We don't tend to put a lot of money into it. Because venison is farmed and it's hunted and it's a food source across so many different areas in the world, people are starting to look and people are getting worried. So the way that you research it is quite complicated. A recent study came out in end of last year or beginning of this year, where they grew miniature human brains. So they're called a brain organelle. So it's the closest thing that we can get to growing a brain in a lab. It's not AI, don't worry, it's not going to take over the world. But when they tried to expose those, for instance, to chronic wasting disease, it didn't pick up. So they're doing some really impactful, very specific in vitro lab work. But at the same time, if they put human prion proteins into mice, they can infect them with CWD. So there's definitely looking, but there's no clear yes or no yet. And hopefully the more worried people are, the more money they will invest in this research and the better and clearer an answer we will get.
That's really interesting. Now, when I chatted with Kate once previously, so the cannibalism in that, and I can't remember the country, what was it called?
Papua New Guinea in the
Papua New
Pacific.
Guinea. Okay, thank you. Yes. So in Papua New Guinea, if I remember correctly, and this might be wrong, the men were eating the muscle, the women and children were eating the organs, and then the time that it took for the disease to become symptomatic was faster when it was the organ consumption versus the muscle consumption. Have I remembered that correctly?
And in particular to note is that women and children were, it was more customary to eat the brain tissue as well. So as part of a funerary procession in that culture, in order to release the spirit and provide the spirit back into your ancestors, you would consume the body as an honorary approach. So the organ and brain tissue did have a much quicker shorter, I should say, incubation time. So children were seen symptomatic with kuru is what the disease was called, whereas the men weren't finding that they were getting symptomatic anywhere near as long a time, shorter time. Took them longer, I should say.
Yeah, I find that really interesting because I guess I can kind of, even though there's been no conclusions on bucks, when you think about their behavior and what they sniff and what they touch and all of that sort of stuff, potentially they could just be being loaded with prions perhaps faster than than a doe would, because she's probably not sniffing all of those other things,
Yeah,
right?
and definitely, and different, bucks tend to fight with other bucks. They also may mate with multiple females, whereas you don't, a one female may only interact with one buck in a season.
Yeah.
But yes, the likelihood of having a prion load higher is much higher in males than in a random doe.
So I have a daughter that has oral allergy syndrome. And so for people that don't know, she's allergic to a broad number of pollens. And it's really specifically the proteins in those pollens that she's allergic to. Then she developed oral allergy syndrome, which is proteins in raw fruits and vegetables that are similar to those proteins. But now for Caitlin, the solution is altering the protein with heat or processing of some kind. Why are these prions so resistant? And if you had, so if it's prion A that's normal and prion AB, because I'm not scientific, AB is the same prion, but now it's misfolded. Is prion A, the normal prion, altered by those things? Do you kind of get where I'm going with this? Like I'm absolutely shocked that we can't destroy these little buggers.
Yes. Yeah. And is the question you're asking kind of, we know that you can't get rid of or destroy misfolded prions, but can we destroy normal prions?
Yes.
From my
Yes.
understanding, yes.
Okay.
And Kate, jump in if that's not correct, but from my understanding, you can.
Yeah, yeah, no, that is correct. And there's something because normal proteins and normal functioning prions are actually quite sensitive to heat and will be altered. But there's something, and again, I don't know if the science community fully understands this, but I have read, multiple resources that say something about that misfolding, that really tangled up slinky, makes them super resistant to heat, and so they lose that. When exposed to heat, they don't break down, and I think that also kind of just plays in generally with because normal proteins in the body will kind of the natural function, they kind of break down to their component parts and reform, and they carry out their functions, but these ones, they don't break down, and that's why they accumulate in the body, right? And then they cause that damage to the central nervous system tissue. So yeah, that's a really talk about a pathogen that has evolved to just be indestructible, right? Like it having that resistance to heat just means that they are like super resistant and persistent, in the environment. So it creates a huge challenge for somebody that's trying to um, manage that. But it's, it's a very successful strategy for the pathogen.
Yeah, I find that really remarkable. So the other thing that that I was curious about is just Kate has given us some insight into, we are now starting to understand that these prions can even be a little bit different from, say, Alberta's prion may be folded a little differently than a Texas CWD prion. Or do you have any information around that sort of thing? And is that the prion evolving? Because I think of COVID-19-1, and we're probably now at COVID-19-101, which I realize is a virus and not a prion, but that evolution is interesting. And I guess part of it is that how effective is a protein, that just simply kills its host all the time, like a bad protein. Do you know what I mean? And so yeah, so I'm kind of curious about that relationship.
Yeah, so I will just start immediately just to say that they don't they don't have a genetic material, a protein, so they don't tend to evolve in the same way as a bacteria or a virus. What does change is the variations you get within the gene that produces prions. So the prion itself, how it misfolds, no one seems to quite know what causes the misfolding. But I don't know if you remember in that first slide that I showed you, there's a normal prion protein, and then you get something called a beta helix shape, which is the misfolding. So it's effectively going from being like this to kind of being like weird and wonky. So the amount of weird and wonky can look different, from my understanding, across different regions. So if you imagine a slinky, you can pull it out. If you twist it here, it's going to not work very well. If you twist it down here, it's not going to work very well either. So in different places with different isolations, you will find that there are changes in the way that prion misfolds, which may be related to that gene, but that's not super clear. But you get that geographic variation because if you've got a pocket of chronic wasting disease, let's say in eastern Alberta near Saskatchewan, that is going to, over time, with a slinky that's twisted down this end, hit other prions and other animals that are also going to twist down that end. If it misfolded spontaneously in another area slightly differently, that's going to look slightly different as it spreads across the West, wherever that might be, or if it spreads in Korea, for example, or in caribou or reindeer in Norway, for example. But it's not evolving in the same way as a bacteria or a virus, which will go into a host, change its genetic makeup to make it more hardy and more aggressive, like COVID,
Yep.
and then move into a different person, evolve again. So different function, similar outcome in that it does have some variation.
So that's kind of interesting. So if I'm following this, then I guess my question would be, is a mule deer in Texas is going to be, which, okay, let's say a whitetail in Texas is going to be is going to have like a certain gene pool. And yes, we have the same whitetails, the whitetail species broadly in, say, Saskatchewan, but genetically there are differences in them. And yet I'm thinking that if a whitetail in Saskatchewan that may be genetically different than to some degree than Texas, but that whitetail with that misfolded protein over on this side then spreads it to the elk, and that misfolded protein is likely then still going to be a similar.
Yeah, so that's the theory, is that if the protein is misfolded in a specific way, whatever it touches will misfold in the same specific way. That
Okay.
being said, different groups of animals, so geographically split. So the whitetail you mentioned in Texas, that's been there for, I don't know how long whitetails have been in Texas, but let's say a number of hundreds or a couple thousand years over time, compared to those in the Pacific Northwest, they will have different genes, right? So there will be differences that have accumulated over time just because the diet is different, the environment's different, the predation is different. And what we do find is that there are variations within the gene that codes for the prion protein. So different genes have different what are called alleles. So that is like variations on the gene. And some variations provide more likelihood to either be resistant to chronic wasting disease. So when challenged with chronic wasting disease, they are more hardy against it. And there's more research looking into, can you be more likely to be misfolded as a prion if your coding gene has another kind of variation. So there's a lot of variation both in how that prion misfolds, but then also how it's coded within your genome if you're a deer.
It's exceptionally complex. So,
Yes.
and for the audience, because I'm just insatiably curious about this, but I hope that if any of you are thinking of chiming in and asking a question, that I'm not missing a hand up or turn your camera on or interrupt, because you're welcome. So when I start, so I have a horse farm in the Okanagan, And what alarmed me when we first started to get connected, with the provincial committee was hay. Are you the hay? So we were bringing hay from Alberta or hay from Cranbrook, various areas, northern BC, Spokane, all of that kind of stuff. So the movement of hay is pretty significant in North America, and certainly that now we export it, of course, to lots of different markets. Are you aware of any research in, like a research setting where that has shown to be the pathway to infection? I can't
So
remember, Kate, if we had discussed this. It just occurred to me. I didn't know if it, yeah.
feel free to jump in anywhere here, Kate. I don't believe that there's been shown that hay has been a cause of infection, but it is well known and assumed that it could be a cause. So prions, obviously, if you are an infected animal and you're salivating all over the hay that you're eating, or over time prions have somehow got near that hay, you can't get rid of it, heat doesn't destroy it, and you move it, theoretically, yes, that could be a cause or the starting the spark of chronic wasting disease in a new area. There is some technology that is slowly coming out that allows you to swab things and test environmental swabs or environmental samples. So that would no longer require just looking at infected animals' lymph nodes, for example, but actually, could you get a sample of hay, test it before you import or export, for example? But Kate, feel free to add to that.
Yeah, no, that was all great. I would add, because I find this super interesting, and the the sort of understanding of this environmental contamination and the potential for prions to move on plant material, be moved around on plant material, and then be capable of infecting animals from that plant material is relatively recent. Like within the last, 15 or 20 years, like we didn't know when CWD first showed up in Alberta, for example, they didn't understand that mechanism. And so there was a paper published, I think as recent as like 20, it was 2010 or 2015, somewhere in that window, where in a research setting, they confirmed that plant material, particularly like, grasses, hay, things like that, can not only, prions can not only be present on the surface of those plants, but the plants are actually capable, if there's prions in the soil, the plants will take those prions up through their roots and they'll pass through the plant material as it grows, which just blows my mind. And so the paper was demonstrating that like prions can bind to plants, the surface of plants, and also be contained within the plants. And then when animals, like lab animals, consumed those plants, they became infected with CWD. And so this is documented in a research setting now. The challenge on the landscape in the real world to kind of show this, confirm this link is very challenging because we don't have the tools to test those environmental samples. They're coming. There's been a lot of advancement with those tools. We just don't have access to them yet. But there has been some work, a large study with the Canadian Food Inspection Agency that regulates a lot of the captive cervid facilities in Canada, Alberta, and Saskatchewan. They were, when CWD first showed up in the prairies in the mid 90s and early 2000s, they were, had major impacts on these captive populations and the producers. And so really strict regulations came in to regulate the movement of animals between facilities and making sure that everything was tested and healthy. And so that like really tightened up, but then they were still seeing introductions in farms when there wasn't any link to animals moving between them. And even like very recently in the last couple of years, there's been a bit of an uptick in new farm properties having new animals testing positive for CWD. And so the Canadian Food Inspection Agency, or the CFIA, did a review of all these cases, and again wasn't able to test the soil and say yep, or test the hay rather, and like yep, there's prions in this hay, but through sort of a process of elimination, they were like, this has got to be the source, basically. And so there's building evidence now that that's not just possible but probably happening. When it's hay moving across the landscapes and into different places, it's very challenging, and it's something that's been on our radar in BC for a long time, and something that we don't have a great handle on because we don't have any authority to regulate the movement of hay, and we don't have these tools to test the hay at the border. So we've really had taken sort of an education position with just trying to increase awareness around this risk, because we know it's certainly possible, but yeah, there's I would say there hasn't been a ton of evidence to say, oh yeah, this is a situation where this definitely happened, but I think I think that's starting to build, not with those direct linkages maybe, but sort of through this process of elimination, saying, this is a likely mechanism of how it's being introduced to new areas.
With my experience on the farm with hay, having brought in a lot of hay over the years, we've had baby deer in the hay. We have certainly had, in almost every instance, deer manure and that sort of stuff. Now, at our facility, we primarily feed grass hays, which the Peachland residents don't beat down the doors for. They will beat down the barn doors for the alfalfa that's in there, and of course that has certainly come with all of the same things. But yeah, it's interesting because of course then that goes through the horse, and then it's out in the pastures, and and the deer will spend time on that green grass, and
Yeah.
it's just, it feels a little bit overwhelming with where it can go and how it can get there, and all of that kind of stuff. So, and in summarizing, I think one of the things that I really liked too with your presentation, Jess, was just that up to 36 years or 36 months, sorry, from exposure to the start of symptoms, and then within a year after that, or around a year, a cervid is dead. So we have potentially animals on the landscape that are living for 4 years, that are sharing and spreading and carrying the disease around as they go. That's a fairly long time. Imagine if COVID had taken 4 years anyways, just, I mean, because of course that's the most recent pandemic that we had in humans. Not the same, obviously, but it just, yeah, 4 years, that's a long time. It's a long time for an animal that can potentially be on the land spreading the disease.
Yeah, it's a long time to be infectious, but it's also a very short time compared to a normal lifespan of a deer. So mule deer, I think, in a normal environment, if they're healthy, they can live 11, 12 years. So if you imagine for a doe, how many fawns it can have in that lifetime, and for males, and bucks, how many they can sire. Is that the right word? Sire? So that's really important in terms of having deer on the landscape. But when we think about that a male deer may be born, there is some links to show that chronic wasting disease can be spread vertically. So infected mothers can give their babies in utero chronic wasting disease, and the babies are born with CWD, which is really sad. But that means a male, for instance, may be living 2-4 years. That's not very long compared to a natural life cycle, and how long the animal would normally live for. So we do see over time significant population declines. Talking with some colleagues in Alberta, they have mentioned that in some areas there are really big declines in bucks in high CWD areas where, you know, guiders are going out looking for bucks with clients and not finding any. That's not a great life. Not great for the environment, not great for hunters, not great for the indigenous people that are using deer as sustenance, but also same for hunters that use it for food security. There's many concerns there from both an ecological and a human safety point of view.
Thank you very much. I recognize that this is always what we aim to do, particularly around consuming, a known CWD positive. It's choice. It's up to people what they find is acceptable for risk and that kind of stuff. And I really love just being able to sort of bring this human element in of if you ate a CWD deer last hunting season, and you're still here, yay. But potentially there are just so many things that are going on, not the least of which is just the evolution. And so, people need to make those choices knowing, of some risk, right?
Yeah, and noting as well
Question.
that you noting as well that you won't know until it's too late. And I think that's a key piece, is that you might eat a number of CWD positive deer and you are fine in 3 years. You're like, oh, they were complaining about nothing. But 4 years from then, 10 years after, you're dead within a year, potentially, if it is transmissible. And we
Yeah.
don't know, and we might find out through science within that 10-year time span. But you really do have to ask the question, is that worth it? If you've got kids, do you want to be, and again, we can't say don't eat CWD deer because there is no evidence to say that it shows or that it is transmissible across the species barrier. But you also don't really want to be the first to prove that it can cross the species barrier, So it is a
Yes.
personal choice, but given that there is so much work put into the testing program, some of those turnaround times, in the management unit are really speedy. Again,
Yeah.
I don't personally think the risk is worth it, but it is up to people to make that decision.
Yeah, for sure. For sure. We have a quiet audience, so we are going to thank everybody that's shown up tonight, and Jess and Kate, as always, we just love having you guys, and Jessica, thanks for making the time for us, because it's these resources that we continue to try and place out there for people to just learn and to understand, new hunters coming in, that kind of stuff we feel is super important, that people make good informed decisions, and you've definitely helped us advance that initiative. So super appreciated.
You are very welcome. And the more information we can get out that's real and not people just like chatting on social media and making
Yes.
scaremongering posts or saying that it's not a
Yes.
problem, happy to help.
Yes. Yep. We love it. We love it. All right. So no other questions from the audience. We're going to say thank you very much. Appreciated you coming tonight, and see you all in the next one.
Sounds good.
Great.
Thank you.
Thanks so much.